Hi, my name's Annie Bryant, and today I'll be walking through this recent paper in Network Neuroscience from my group at the University of Sydney and our collaborators at Monash University in Melbourne. And it's titled Benchmarking Overlapping Community Detection Methods for Applications in Human Connectomics. I've highlighted our fantastic team of co-authors, and I want to specifically shout out Dr. Aditi Jha, who's now a postdoc at Stanford and is my fellow co-first author on this paper. The core idea for this paper rests upon community detection algorithms, which leverage principles from graph theory and network science to characterize the inherent architecture in the brain's connectome, be it structural or functional. And this is specifically a graph theoretical method based around modularity in the human brain, meaning the extent to which nodes, which could be regions or voxels, are grouped into distinct communities based on their connectivity to other nodes. And community detection algorithms are used to infer the structure of these modules, which can be mapped back onto the brain to interpret biological significance, such as comparing functional integration versus segregation over time. And in this work, we'll be focusing on the structural connectome estimated from diffusion tensor imaging, which can be represented as an adjacency matrix, where the rows and columns are regions, and each cell captures the estimated streamline count. And this can in turn be represented as a network of pairwise connections. And the standard approach for community detection has historically been the Louvain algorithm, as described by Blondel and colleagues, which applies hierarchical agglomeration to optimize the modularity of the resulting communities. And this algorithm, as well as the related Leiden algorithm, yields discrete, non-overlapping modules, and it's been the most widely used method for community detection in network neuroscience. I want to highlight this aspect of the Louvain algorithm, which is that the resulting communities are discrete and non-overlapping, meaning that each node is assigned to a maximum of one community. We know that the brain exhibits a spatially distributed and fluid network of complex interactions, which can be an issue when community detection imposes a static, non-overlapping modular decomposition. And it's important to efficiently capture and interpret this overlapping community structure in the connectome to advance our knowledge of how the brain integrates information across distributed networks. Recognizing this pressing need for algorithms that can flexibly capture the overlapping architecture in the brain, there's a growing repertoire of overlapping community detection algorithms, or OCDAs, at our disposal. To date, most applications of these methods have been to the functional connectome, with only a few studies looking at overlapping architecture in the brain structure. In humans, this has primarily comprised gray matter covariance networks by Wu and colleagues, and subsequently by Sotiras and colleagues, who also looked at structural connectivity in mice using diffusion tensor imaging. More recently, Armas and colleagues examined overlapping community architecture with retrograde track tracing in the macaque brain. However, these studies and other applications to the functional connectome have generally started with the manual selection of a particular OCDA, meaning that brain imaging studies across diverse and important applications could be relying on suboptimal subjective choices that might obscure the most relevant overlapping modular decomposition for a given connectome. Moreover, each of these algorithms makes different assumptions about edge weights. For example, click percolation and SLPA binarize edge weights, while OSLOM and NMF and InfoMap inherently incorporate the graded structure of edge weights. And this divergence has important implications for benchmarking and requires careful consideration of how, and indeed the extent to which, edge weights could affect algorithm performance. Additionally, while there are many variants of these algorithms and accordingly many options for parameter configuration, we opted for implementations with only one, if any, user-defined parameter to systematically vary such that all other conditions were fixed per algorithm. And so together with co-first author Dr. Aditi Jha, who I mentioned at the start, we aimed to address this gap through a systematic algorithmic framework centered on the right hemisphere structural connectome, summarized across 973 participants in the Human Connectome Project dataset. And I'll unpack the details shortly, but the gist is that we implemented a generative network model tuned to attributes of our structural connectome to simulate 1,000 benchmark networks with known ground truth overlapping architecture. We then applied each of the above algorithms to this ensemble and a variety of their parameter configurations. The upshot is that this offers a data-driven approach to quantitatively identify the optimal OCDA for our target empirical network. The generative network model that we used was developed by Lancic-Canetti and Fortunato in 2009, which yields weighted networks with known ground truth overlapping community assignments. And these simulated networks are constrained by parameters estimated from the empirical structural connectome, either directly or via power law fitting algorithms, as well as some parameters sampled from manually defined ranges. And so here's the group averaged structural connectome from diffusion tensor imaging, where we have retained only edges with the lowest 15% variance across individuals. We aggregated across all 180 regions in the right hemisphere Glasser atlas to compute the regional strength as the sum of all streamline counts to other right hemisphere cortical regions. And we confirmed that nodal strength and degree are indeed proportional as expected. One important caveat is that this generative network model imposes a scale-free power law distribution, which is not observed here, and indeed often is not observed for many real-world networks. So we hope that future work might incorporate a larger range of topological constraints and expand beyond power law distribution for degree to enable more precise calibration of the suite of OCDAs to a given target empirical network. But I'll digress for now and just quickly review the key parameters governing the stochastic network generation algorithm that we used here. So I've color-coded these parameters into three categories. Those with a fixed value set to directly match the empirical connectome are in green. Those with a fixed value estimated from a power law fitting algorithm are in pink. And those sampled from a specified range that we manually titrated are in blue. And so I'll first introduce the concept behind each parameter before showing the actual values we used in our model. First, we fit a power law to the empirical degree distribution of the structural connectome, estimating the scaling exponent tau sub 1 while fixing the mean and maximum degree to match the observed network. Edge weights are then assigned using a weighted formulation in which node strength scales with degree according to a power law relationship governed by the parameter beta. Community sizes are sampled from a second power law distribution with scaling exponent tau sub 2, with manually specified minimum and maximum community sizes and parameters governing the number of overlapping nodes. And finally, two mixing parameters are sampled. Mu sub t controls the fraction of a node's connections outside its community, while mu sub w controls the fraction of its total edge weight external to its community. And so here I'm showing the set of values for our fixed parameters, either fixed directly to match the empirical structural connectome in green, or estimated from the power law degree distribution fit in pink, or else randomly sampled from the specified ranges chosen to sweep a reasonable range of plausible empirical values. Okay, so now that we've covered how the ensemble of benchmark networks is simulated, let's look at how we evaluate performance of our suite of OCDAs on this benchmark ensemble. We chose to use the same measure for performance evaluation as in Lancic, Canetti, and Fortunato, which they termed the extended normalized mutual information. I'll briefly unpack this measure here, but I would refer the interested reader to Appendix B in their paper for more details. The core building block of this measure is mutual information, which is a symmetric measure of the average information that the value of one distribution, X, which here represents the ground truth community structure, provides about another, Y, which here represents the algorithm predicted communities, and vice versa. Mathematically, it's defined as the difference between the sum of the marginal distributions of X and Y minus the joint entropy of the two distributions together. In 2005, Dannon and colleagues expanded upon this measure to describe the normalized mutual information designed specifically for community detection, which we represent schematically here as the quotient of the mutual information between X and Y divided by the average of their respective marginal entropies. The resulting quantity ranges from 0 to 1, with 0 reflecting completely independent clustering. For example, if an algorithm assigned every single node to one cluster, up to 1 reflecting perfect agreement in cluster labels between the ground truth and the algorithm. And so Lancic-Ganetti and Fortunato further carried this measure forward to introduce the extended normalized mutual information, specifically to allow for multiple community assignments per node, using binary encoding of community assignment to capture the random variables boldface x sub k across all communities in boldface x. This involves computing the conditional entropy of each community x sub k across all nodes and normalizing it across all community assignments. And again, further details beyond this summary can be found in Appendix B of the referenced paper. Okay, so now we know how to simulate an ensemble of 1,000 benchmark networks, apply our set of 5 OCDAs and their parameters to this ensemble, and evaluate how well these algorithms can recover the latent ground truth overlapping architecture of each simulated network. So here I'm showing one example simulated benchmark network from this ensemble. And this colorful tapestry reflects the community assignments from the ground truth in pink and from the evaluated OCDAs and their corresponding parameters in black. Across the 1,000 simulated connectome-like networks in our benchmark ensemble, we found that the Oslam algorithm best recovered the overlapping network architecture. This is a local method for community detection that assigns clusters and evaluates how likely or unlikely it is that a given cluster has formed by chance. We examined different p-values for the cutoff of the significance threshold. We progressed with this method using a p-value of 0.3 for the rest of our analyses. For robustness analysis, we also confirmed that the Oslam algorithm struck an optimal balance between sensitivity and specificity of recovered overlapping node identity in particular. So our implementation of Oslam to the right hemisphere structural connectome yielded 7 communities that were anatomically and functionally specialized and primarily spatially contiguous. Within these 7 networks, Oslam identified 15 overlapping nodes, 14 of which spanned 2 communities, and one of which, the retrosplenial cortex, spanned 3. And these 15 regions do indeed exhibit cross-module structural connectivity, and they're also densely connected to each other, which hints at an integrative bridging structure across the distributed networks. Another interesting note is that the overlapping regions generally sit at the spatial interface between the 2 or 3 networks to which they were assigned. Comparing the alignment of our 7 structural networks to the 7 canonical resting state functional networks from Yao and colleagues, we found that about half of the overlapping nodes mapped primarily to higher order default mode and frontoparietal or cognitive control systems, which comprise association and paralimbic cortices, while the other half of regions correspond primarily to the visual network, which is a unimodal system, or the ventral attention and limbic networks, which are heteromodal systems. And we thought this might reflect a natural bipartition into nodes at opposing ends of the putative functional hierarchy of the cortex. So we linked our OCDA-defined brain maps to the first principal gradients of functional connectivity, which reflects an intrinsic axis of hierarchical variation in the cortex. And indeed, regions that were found to overlap across multiple structural modules sit higher in the first principal gradient overall, and are found entirely along the visual to transmodal axis. This is more of an aside, really, but I also just wanted to point out that the overlapping regions have an overall higher node degree and are also statistically larger than non-overlapping counterparts on average. The Oslam-defined community structure also offered more nuanced nodal statistics than the standard Louvain decomposition. So here we've plotted the participation coefficient capturing cross-module connectivity, versus the within-module strength, capturing intra-module connectivity for the Louvain decomposition. And these colored-in dots reflect regions that Oslam judged as spanning multiple modules. We expected these regions to show a greater spread of connectivity to nodes across multiple Louvain-defined modules, in which they would sit closer to global connectors or at least to provincial hubs. And if we compare the change in participation coefficient with the overlapping Oslam versus the non-overlapping Louvain, we see that amongst a backdrop of overall increased participation, two nodes stand out in particular, which are 9M and 8BM, both at the anterior medial surface of the cortex. Zooming in on the communities to which Louvain versus Oslam assigned these regions, we see that Louvain assigned them both to one larger community, while Oslam more flexibly permitted separation into three distinct communities, collectively bridged by these overlapping regions. And robustness analysis across different randomization seeds and values of the gamma parameter for Louvain supported the same conclusion. We continued by computing the participation coefficient of our 15 overlapping nodes across the communities identified by Louvain versus Oslam and compared the values with that of the first principal gradient of functional connectivity that I introduced before, finding that only the Oslam-based participation coefficient was significantly positively associated with the first principal gradient. And this suggests to us that overlapping regions identified by Oslam exhibit greater cross-module intrahemispheric connectivity, which in turn positively correlates with their position in the putative functional hierarchy of the cortex. And indeed, a similar pattern held when we looked at all 180 regions in the right hemisphere Glasser parcellation. Although our application here was to structural connectivity rather than functional, I do think this ties in with really interesting work from Janine Biesterbosch and colleagues at Washington University St. Louis, showing that regions participating in multiple functional networks are more likely to exhibit linear additive coupling as integration hubs, routing communication across otherwise distributed networks. So with that, I'll end there and thank my collaborators on this work, particularly co-first author Aditi Jha and my thesis supervisor Ben Fulcher, along with lab members from my thesis and now postdoctoral work. I'd also like to thank the funding bodies that generously supported this work and all other projects throughout my thesis. And most importantly, I'd like to thank you for taking the time to watch this video. Please feel free to email me if you have any questions, concerns, or feedback about this work or my presentation of it here.